delighted today to introduce Dr. Myron Evans. He is currently a postdoc at St. Jude's in the Hatley lab, but he is very soon going to be starting his own lab at the University of Washington here in October, so keep your eyes open for announcements for hiring and the such, so it's going to be very exciting. And given his fantastic background, I'm not surprised he landed such a great position. And so I wanted to mention Myron has won numerous awards, including Basic Science Scholars Award with the American Association for Cancer Research, lots of poster awards and travel awards, special fellowships, like lots of accolades for his fantastic research. And he's been doing a lot of really cool work on understanding how PRC2 is involved in neural development. I'm really excited to hear Myron speak today about his work, and maybe we can get a glimpse of what the lab's going to be like starting in October. So thank you so much for joining us today, and I will stop talking.
Absolutely. Thank you so much, Christine. All right, so today I'm going to talk to you guys a little bit about a paper that I worked on that was actually got published in the middle of quarantine, but because of it sort of being, everybody was in their house, I never really got to talk in an open seminar about it. But we identified this transcription factor Ybx1 as a novel regulator of polycomb function controlling embryonic brain development. And so when I joined Jamie's lab, my first postdoc here at St. Jude, we sort of wanted to answer one really basic question, and that's how do cells acquire and maintain their unique identity and function? And so one of the things that we know about development is that it's a restrictive process where you move from a totipotent zygote through pluripotency down to what we consider maybe a unipotent state or a no potency state, where the majority of the organs in our body have some stem cell activity. But much like organs like the nervous system have very little to no stem cell activity and display no potency. And this restrictive process is mediated by changes in chromatin accessibility and lineage potential and speciation of different cell types, as well as transcriptional permissivity. But sort of at its driving event, one of the things that we know is that epigenetic regulation by both DNA methylation and histone methylation is extremely important in driving this, these cellular phenotypes. And I'm not going to talk about DNA methylation because it's been pretty well studied, but I'm going to talk about histone modifications and how we've identified novel regulators of those. And so I think it's very well known by most of the people in this group that the combinations of these DNA modifications and histone modifications are what drive your chromatin state and allow us to identify certain regions of the genome. So you can see here, as an example, if you have this combination of K4 trimethyl, monomethyl, and K27 acetyl, you can identify that as an enhancer. You can also identify active promoters, poised promoters, silent promoters, as well as actively transcribed genes and gene bodies. And in our lab, we've really focused on one particular set of these modifications, and that's what drives this repressive state, and so H3K27 trimethylation. And one of the reasons that we really focused on that is sort of twofold. So my background prior to joining Jamie's lab was in cancer, and the two regulators of K27 trimethylation are both mutated in numerous forms of cancer. And also, it's well known that this repressive state is actually required for genes to be activated. And so you actually have to lay down the K27 first, and in order for it to be removed, the enzyme that removes it also happens to activate transcription and translation. And for years, we've known exactly what regulates K27 trimethylation genome levels. And so you have the polycomb repressive complex 2, or PRC2, which serves to catalyze the methylation of lysine 27 on histone H3 and leads to compaction of chromatin and blockade of gene transcription. And most commonly studied UTX, but also JMJD3, which removes the methylation marks and allows for activation of gene transcription by interacting with the machinery for methylation of lysine 4 on histone H3 and allowing for gene transcription that way. And one of the questions that I had when I sort of joined the field, I didn't have an epigenetics background, I didn't really know that much about epigenetics, was, well, how does PRC2 know where to go? All the DNA in every cell in your body is the same, but very clearly we know that H3K27 is deposited in specific regions of the genome and specific cell types. And so when polycomb was first discovered in Drosophila, it was very computational analysis went on to show that we could find these polycomb responsive elements, or PREs, throughout the genome, and wherever you find a PRE, you find polycomb. And elegant work by people in the field have actually shown that you could pick up these PREs and move them to new regions of the genome, and polycomb would just sort of follow along. where those PRE elements. So sort of showing that in Drosophila and in invertebrates, polycomb functions in a responsive model. So wherever PREs are, polycomb is there. And when we thought for a long time that this possibly wasn't the same in mammalian systems and hoped that we could find something that was a more instructive model. And so computational biologists sort of looked for these PREs in the mammalian genome, and we couldn't really find them. We could find polycomb responsive element like regions of the genome, where we commonly see PRC1. And as of now, and at the time, one of the things we could only see was that PRC2 was commonly found in CPG islands adjacent to promoters. And so we thought that there might be an affinity for these CPG islands. Interestingly enough, approximately 70% of promoters in the human and in the mammalian genome are flanked by a CPG island. So there's also the possibility that there just happens to be CPG islands around promoters, and that's just where you see them. And so our lab was really interested in trying to understand if there was a possibility of an instructive model. And about 11 years ago, when Jamie was a postdoc, she published an elegant paper in Cell that was actually sort of bound by 4 other papers that were published in Nature Cell Bio and a few other journals that identified this novel interactor of the polycomb repressive complex 2 called JARED2. And if you look, JARED2 actually does have an affinity for GC-rich regions of the genome, so that may also explain why we see it at CPG islands. And in multiple of those papers, they went on to show that knockout of JARED2 was deleterious for PRC2 recruitment. So here's shown in mouse embryonic stem cells on the left in the Lindera et al. Paper. And in Jamie's paper, she showed that morpholino injection of JARED2 in Xenopus mirrors the PRC2 injection, PRC2 morpholino injection, inhibiting gastrulation. And so really showing that JARED2 may have a function that's similar to PRC2 and is extremely important for development. And over time since then, we've identified a number of other interactors of PRC2. And we now know that PRC2 actually exists as 2 completely separate functional complexes. So we have PRC2.1 that's characterized by the core subunit, CZ12EED, and either EZH1 or 2, interacting with PCLs 1 through 3 or PALI 1 and 2, as well as PRC2.2, which interacts with JARED2 and another accessory protein called AEBP2. And although there's significant overlap of where these bind, their functionality is synergistic in both mouse embryonic stem cells and human embryonic stem cells. And so we were interested in understanding, well, if JARED2 doesn't completely lead to deletion of PRC2, and neither does deletion of PCL1 and 3, are there possibly other interactors that hadn't been identified yet? We were really interested in this because at the time when I was joining the lab, CTBB1 and 2 had been identified. And they were only able to be identified by looking at interactors of PALI 1 and 2, because if you looked at interactors of the core subunits, CTB1 and 2 were so far down the list that you might have assumed that they weren't actually functional interactors and had nothing to do with the complex themselves. And so we decided that maybe we should try to undertake that with JARED2, since Jamie had been sort of on the discovery side of that. And so our thought process was that maybe there was a transcription factor or a non-coding RNA that may more strongly interact with JARED2 rather than the whole of the PRC2 complex or the core subunits, and may help decide where PRC2 is bound around the genome. And that may help us answer the question of how does PRC2 complex sort of achieve this dynamic gene regulation. And so we started in human embryonic stem cells and performed an IP mass spec analysis of JARED2 to see if we could identify any novel interactors of JARED2 that would have also affinity for PRC2. And this is the data shown here. And so you can see that if you IP JARED2, the bait comes down really well. You can pull the other subunits of the PRC2.2 complex, including ABP2, but we never pull any of the PRC2.1, showing the specificity of the complex. But we also grabbed this novelly identified protein called YBX1 that hadn't been shown before to interact with either JARED2 or the polycomb complex. An IP Western blot in human neural progenitor cells actually showed that, in fact, JARED2 and YBX1 interact pretty strongly, typificated by here, but JARED2 IP in neural progenitor cells. And if you actually IP for YBX1, you can still pull down JARED2, and you'll see that we don't pull down as much JARED2 as we do if we IP for JARED2 itself. And we think this is mostly a stoichiometry issue, that there happens to be way more YBX1 in a cell than there is JARED2, and YBX1 interacts with other proteins. And again, you can see that here, if you IP with CZ12, we do pull down some YBX1, but it's a very, very small amount. And so we think that for earlier IP mass specs, this may have been missed. The interaction with JARED2 and YBX1 is a protein-protein interaction, so it's not mediated by DNA or RNA input. So we treat the cells with benzonase, or the nuclear extracts with benzonase, or we do bacterial extracts. We can see that JARED2 and YBX1 directly interact with each other. And so what is Ybx1? So it's been really heavily studied in oncogenesis. It's a DNA and RNA binding protein that contains this catalytic cold shock domain here. And it's been shown to be involved in both DNA repair and replication, as well as functions in the cytoplasm. And you can see here on the schematic that it has both a nuclear localization signal and a cytoplasmic retention signal, and so it can shuttle back and forth between the cytoplasm and the nucleus. And so our pipeline in the lab, when we identify these interactors, is to knock them out in human embryonic stem cells and see what happens to them. And we did this before on UTX, where we identified an interaction with 53BP1 and showed that loss of 53BP1 leads to a differentiation defect in human embryonic stem cells into a neural lineage. And try as we might with YBX1, we actually couldn't get a knockout, and that's because we think YBX1 is required for pluripotency in human embryonic stem cells, which will be something that I'm working on in the lab when I start it for myself. But luckily for us, there happens to be a mouse model of YBX1, and the mouse YBX1 knockout and the mouse and human versions of YBX1 are 97% homologous. So we can confirm that, in fact, the mouse version of JARED2 and YBX1 interact, shown here, that these knockouts don't express any YBX1 protein, and that loss of YBX1 doesn't significantly downregulate JARED2 expression. And so what about these animals, these knockout animals? And so they have a characteristic neural tube defect that's similar to those seen in haploinsufficiency of Z12 or deletion of JARED2. And so we really wanted to try to understand, well, what's the effect of YBX1 on the neural tube and its development, and why do they have these defects? And so I'm going to blow through some of this data really quickly. If you have questions about it, we can talk about it at the end. But what we ended up showing is that, in fact, knockout of YBX1 leads to increased proliferation, measured here by FACS analysis and by IHC staining. And that also that increased proliferation leads to a loss of differentiation capacity in these cells. And so YBX1 knockout cells cannot differentiate into neurons, shown here by TUJ1 staining and quantified down here on the bottom. And then we further went on to show that YBX1 actually has to be in the nucleus in order to exert its effect. And so when you delete the nuclear localization signal, you can't actually reverse the proliferation defect, whereas you can in the full length when you replace it with the full length version of YBX1. And so in the nucleus, we know that YBX1 binds to DNA. So does it actually lead to altered transcription in neural progenitor cells? And in fact, it does. And that's shown here in the volcano plot. There's a number of genes that are both downregulated and upregulated. The downregulated terms tend to associate with neuron projection development or neuron development, which isn't surprising, whereas those in the upregulated side tend to correlate with proliferation and cycling genes. And so this is just a quick summary. I'm going to keep moving ahead really quickly. But we have identified this YBX1 as this interactor of PRC2 and JARED2. We know it binds DNA, but where does it bind? Do those sites overlap with PRC2? And so using the cut and run technology that Steve Henikoff's lab developed, we found that YBX1 tends to bind promoter proximal, either 3' or 5' to the transcription start site, with about 20,000 peaks around the genome. That its expression correlates with downregulation of gene expression, although kind of weakly, as shown here on the right. And again, those downregulated terms drive nervous system development, whereas those on the opposite side tend to relate to polycomb by regulation of transcription by RNA polymerase II, or Hox genes, which are characteristic PRC2 targets. And so what happens to PRC2 itself when you remove YBX1? And so strikingly, surprisingly to us, if you remove YBX1, we actually see a global increase in K27 trimethylation. And that's shown here by both cut and run on the left and ultra-low input native chips, the two different technologies that sort of show us the same phenotype, and that you get a global increase in K27 trimethylation, and there's no alteration in PRC2 components either by RNA-seq or by the protein level. And so really showing that loss of YBX1 seems to lead to this hyperactivation of the polycomb complex. And if we sort of take a dive into some of those neurodevelopmental genes, first you can sort of see on beta actin, we don't see any binding. This is really just to help us with our specificity, and no alterations here. But on genes that are downregulated upon loss of YBX1 that are required for neurodevelopment, we see a significant increase in K27 trimethylation promoters of these genes, which most likely leads to their silencing. And so it seems as though loss of YBX1 leads to this hyperactivation phenotype for PRC2. And so what about PRC2 binding around the genome itself? And so what we ended up seeing was that, surprisingly and strikingly, when we lose YBX1, we actually lose PRC2 binding, as measured here by EZH2 and EZH2 cut and run, in most of the regions around the genome. And you can really see that here at FOXG1 and NeuroD2 again. And so loss of YBX1 in the blue leads to a decrease in EZH2, although we do see a concomitant increase in K27 trimethylation. And our computational biologist, luckily for me, is very good at analyzing this data. And what they think is that although we see a lower level of EZH2, It's that the peaks aren't very high, and so the called peaks shown here are much lower, but we see a spreading of EZH2 throughout the genome, and so we actually have more EZH2. It's just at a slightly lower level, and it's not enough to pick up on the peaks when you sort of run the computational programs, and that's why we see an increased K27 trimethylation. And we can also see that if we look at gene deserts like this one on chromosome 3, we start to see EZH2 get placed into random regions of the genome. And so it seems as though YBX1 is both required for the suppression of PRC2, but also localizing it to act to promoters of genes that need to be turned on at poised promoters. And so if that's the case, and loss of YBX1 leads to this increased PRC2 deposition, antagonism of PRC2 should actually revert the effects we see. And in fact, it does. So if we treat our YBX1 knockouts with GSK126, which is a potent inhibitor of EZH2 activity, we can revert the neuronal differentiation defect, and now we can actually get neurons in the YBX1 knockouts. This also reverses the majority of the gene expression defects we see, as well as the proliferation imbalance that we saw before. And in a genetic model where we delete EED using a nested Cree allele, where we deplete most of the H3K27 in neural progenitor cells, we again see a reversal of this phenotype. And so double knockouts for YBX1 and EED look really similar to the single knockout for EED in a YBX1 wild type background. So really showing that we do get a reversal as close to a wild type phenotype as we can. And again, this also reverses the gene expression effects and the proliferation. And so that led us to a really simple working model where we think it poised forebrain neurodevelopmental genes. We have YBX1 bound proximal to the transcription start site, and PRC2 is present there, and that leads to deposition of a small amount of K27. Once signals come in to tell these cells to differentiate, YBX1 and PRC2 and the K27 trimethylation are removed. And differentiation proceeds as we would normally see, and we get a really good balance of self-renewal and neuronal differentiation, as well as forebrain and hindbrain specification, which I didn't talk about, but I could answer any questions about that as well. Upon loss of YBX1, we see this spreading of PRC2 sort of through the gene body in some instances and backwards to the transcription start site. And we get increased density of K27 trimethylation, and despite signals that tell these cells to differentiate now, they continue self-renewing at the expense of neuronal differentiation, and they have an inability to specify their forebrains. And so these animals, but we can again revert this by antagonizing the PRC2 complex. And so, as I mentioned, this was published in Nature Communications in 2020, and this is sort of what I used as the basis for my lab. One of the big questions that I had at the end of this was, well, what about later development? So these animals, as I told you, are embryonic lethal. And so we've created a YBX1 flox mouse model where now we can study later brain development, specifically in the cerebellum. And as well, because YBX1 has been studied in oncogenesis, but we've identified this new epigenetic modification. I'll be looking at its role in epigenetic modification in a few various cancer types, most of them, again, associated with the central nervous system. And so with that, I want to just do some quick acknowledgments to my lab, my former lab, and Jamie for sort of teaching me everything I know about epigenetics and the rest of the lab there. The Hatley lab, which is shown here, which is the lab I'm in right now, where I'm using this epigenetics background and a pediatric soft tissue sarcoma called rhabdomyosarcoma to really help them close out some really cool mouse models that they have of the disease. All the core facilities that I've worked with, and my new collaborator and co-mentor, Martine Roussel, and the funding sources I've had over the years since I've been here. And with that, I will take any questions.
Thank you so much. That was fantastic. We already have a question from Leonie. So if you would like to, I think you can just unmute, and do the camera. Go for it.
That was terrific, Myron. Thank you so much. I'm going to read your paper straight away. It was brilliant. So what sequence does YBX1 bind to? Do we know that?
Yeah, so there had been a reported sequence prior that was what they called a cat box. In our data, we see some binding at these CAAT boxes. We also see binding at other regions of the genome, unsurprisingly. We do see heavy binding of YBX1 in CPG islands as well, which is unsurprising based on the fact that it's interacting with PRC2. And so there may be some specificity at these neurodevelopmental promoters. We tried to analyze that using sort of a motif analysis, and it wasn't statistically significant. And the probability is that just maybe because we pulled the cells out at a specific time, we may need to go earlier or later in development to really nail down that motif. But I wouldn't be surprised if there's something very specific in the brain about neurodevelopmental promoters that leads to YBX1 binding, specifically those regions.
Thank you.
That's so cool. I was wondering, do you think when you're seeing like, I guess fewer or shorter peaks, could that be like PRC like maybe binding and like scanning, and maybe without YBX it's like stabilized, or it's not as stable, but it can still function, I guess. I don't know.
Yeah.
We don't have PRC in yeast, so it's a little bit, I'm not quite on top of the literature, but just curious.
That's a great question. So that was actually my first thought, was that
Oh,
YBX1 is required to actually retain PRC2 at those sites. And so it's not really spreading so much as it's scanning around the genome, and it's like, I don't know where to go. I can't, am I supposed to stop here or here? But we didn't really have a way to measure that. And so after we submitted the paper, I actually did present this as a poster. And somebody came up and posted, like, we have a way to measure this really simply. And I was like,
cool.
we're not going to do it for the paper, but we'll talk to you afterwards. And so now we're in the process of, Jamie's actually looking at that right now to see if we can see if YBX1 is required for motif stabilization of PRC2 at a very particular region of the genome.
Cool, that's awesome. Question from Nicholas Sapp. He says, great talk. Are there any histone modifications that are associated with YBX location within the genome?
Yeah, so obviously K27 trimethylation we can see there, as well as K4 trimethylation. And so it's been shown that YBX1 previously in, I want to say, a fibroblast cell line, that YBX1 interacts with P300 and CBP, and it can alter its functionality. So that would be associated with K27 acetyl. And so one of the things that we're working on now is actually trying to understand what is the interactome of YBX1. We think it's a pretty promiscuous binder, and that it's actually going to interact with a bunch of epigenetic regulators. Whether all of those interactions are as direct as the JARED2 YBX1 interaction, or if those are predicated on binding to regions of the genome that also other epigenetic modifiers bind to, is going to be something pretty important to understand. But I definitely think that, yeah, we're going to, as we start to look more around the genome and move to different stem cells and different cell types, we'll see that YBX1 is going to be associated with other things, and it may not always be K27 trimethylation in other cell types.
Cool. Okay, now we have another question from Anirban. Great talk. I agree. I might have missed it, but do we know if YBX1 has any role of impacting K27? Would it be methylated or acetylated based on the differentiation context?
Yeah, so that is a fantastic question. So we actually did K27 acetyl cut and run when we were doing this. If anybody's done it, it's kind of tough because you use calcium in the process, and obviously marks love calcium. And so the data that we had, we ended up not putting in the paper because it wasn't sort of perfect to where we could really understand what we were seeing. But what we do see is that regions of the genome where we lose or there's a drop in K27 trimethylation, which there are a few, we actually do pick up K27 acetyl. And so there's a possibility that when you lose YBX1, that YBX1 may drive competition between P300 and JARED2. And so when you lose the interaction between PRC2 and YBX1, maybe P300 actually can just come in and start to acetylate those regions of the genome. But that's something that we'll definitely have to use some more advanced technologies to figure out. And I think, again, it's going to be very cell type specific. And so hopefully, once we publish this YBX1 flox mouse and we can let these animals grow to birth, hopefully other people will want to take advantage of the mouse and look in their systems of interest and really start to understand, is this a global thing that we see, or is it very specific to the brain?
Cool. Great. Thank you so much.